So which ions actually flow through the acetylcholine receptor? Turns out that the acetylcholine receptor is a promiscuous cation channel, with conductance for sodium, potassium, and a small amount of calcium as well. You might notice that these are all ions that are cations, or positively charged ions. This is deliberate, since the acetylcholine receptor evolved in such a way that the central pore of the channel is lined predominantly with negatively charged amino acids, from the M2 subunit of each subunit. This assembly of negative charge below the funnel-shaped entrance region in the large extracellular domain of the receptor allows for the attraction of positively charged cations. These cations diffuse through the central pore and exit through the smaller intracellular domain to generate the observed postsynaptic current. Since the acetylcholine receptor conducts both sodium and potassium, it's worthwhile contrasting the effects of this ion flow in more detail. Recall that in a neuron at rest, the concentration of sodium outside the cell is much larger than the concentration of sodium inside the cell. Vice versa for potassium, which shows a much higher concentration inside the cell than outside the cell. Once the channel opens after binding of the acetylcholine ligands, due to its strong driving force, sodium will flow into the cell to depolarize the cell while generating an opposing outward current. But potassium will flow out of the cell while generating an inward current. This process continues until there is no net charge transfer between the inside and outside of the cell. In other words, the inward currents are equal and opposite to the outward currents. For the acetylcholine receptor, this reversal potential occurs at negative 10 millivolts, which is the average of the sum of the Nernst potentials of both sodium and potassium. You can think of this reversal potential as the equivalent to the Nernst or equilibrium potential we've encountered in the previous module for a single ion permeable channels, including the voltage-gated sodium channel or potassium leakage channels. Practically, this means that ion channels which let more than one ion pass simultaneously will always show a reversing potential which intrinsically controls their ability to depolarize the postsynaptic cell and trigger an action potential. We started this lesson by mentioning the need for efficient removal of neurotransmitter molecules from the synaptic cleft to avoid unwanted excitation and inhibition to occur. What if this removal process either fails or is overpowered by too much neurotransmitter being released? In the case of the acetylcholine receptor, and for other ligand-gated ion channels, there's a built-in safety mechanism to avoid unwanted excitation or inhibition. If channels are open for a long time due to the repeated action of neurotransmitters binding and unbinding from the channel, individual channels will close. This process is called desensitization, and it's analogous to the inactivation of voltage-gated sodium channels that we encountered in the first module of this course. The result is the same as if the receptor would have been blocked in the first place. Why is this a good thing? Desensitization of the acetylcholine receptors has proven very valuable in medical practice, particularly as a muscle relaxant during surgery. Succinylcholine is commonly used in surgical practice as a muscle relaxant because it causes desensitization of the acetylcholine receptor. Chemically, it's very similar to acetylcholine and can thus bind to and activate the acetylcholine receptor. Its big advantage, however, is the fact that it can't be cleaved by the acetylcholine-removing enzyme, acetylcholinesterase. As a consequence, succinylcholine can remain in the synaptic cleft for a very long time, ultimately leading to acetylcholine receptor desensitization and muscle relaxation, or paralysis. Interestingly, certain pesticides and nerve gases, such as sarin, employ the same mechanism of action to induce paralysis of the skeletal musculature. In addition, nicotinic acetylcholine receptors can be blocked by certain plant poisons, such as curare. Many man-made pharmaceutical compounds, such as hexamethonium, and naturally occurring toxins present in the venoms of snakes and shellfish, such as alpha-bungarotoxin. All of these compounds act in the same way. They block nicotinic acetylcholine receptor-mediated neurotransmission. As a result, they cause weakness of the skeletal muscles. If administered in a sufficient dose, their action leads eventually to death by asphyxiation due to the paralysis of the diaphragm.